Bismillah ar-Rahman ar-Rahim. The subject today is about immunity, the complement system. The complement system refers to a series of 34 proteins circulating in the blood in inactive form. They can interact with each other to form a series of biological processes. Complement proteins make up about 10% of serum protein. The source of these complement proteins is mainly the liver and the hepatocyte. Uh, the pathways, the pathways of the complement system. There are three pathways of the complement system. First one is the classical pathway. Second one, the alternative pathway, and lastly, the Menin binding lectin pathway. Activation of classical pathway. The activation of the classical pathway occurs by antigen antibody complex, an important and the main activator of this pathway. Also, it's activated by C-reactive protein and cell and plasma derived enzymes like plasmine. Now, the activation of the alternative pathway. The alternative pathway is activated by endotoxin like bacterial lipopolysaccharides, an important activator, also by cobra venom factor, and lastly, cell and plasma derived enzymes. Activation of Menin binding lectin plasma pathway, MBL, this is abbreviated as MBL. Now, by binding of lectin to mannose of microbial cell wall. The mannose of the microbial cell wall bind to lectin, and this triggers this pathway. Now, the activation of the complement system. The complement system composed, as we mentioned before, of three pathways: classical pathway, alternative pathway, and MBL. Classical pathway begins with C1 complement number 1, which is composed of three subunits. C1Q is composed of 6 molecules, C1R 2 molecules, C1S also 2 molecules. Now, this pathway begins with the binding of C1Q to the antibody of antibody-antigen complex. The antibody here is the immunoglobulin M or immunoglobulin G. By this binding, stimulation of C1R and C1S occurs. The stimulated C1S called the serum protease that cleaves the C4 and C2. The C4 is cleaved into C4A, small and inactive molecule, and C4B, large and active. Now the C2 is also cleaved into C2A and C2B. The C2B binds the C4B to form C4B2B. This complex is known as C3 convertase that acts on C3 to cleave it into C3A, small and inactive molecule or fragment, also called anaphylatoxin. Now, another product is the C3B that binds to the C3 convertase to form a complex C4B, 2B, 3B, known as C5 convertase, which acts on C5, again yielding in C5A and C5B. C5A small and inactive, C5B is large and active fragment that attach to the cell membrane of the microbe. Imagine that this is cell membrane of the microbe. Any microbe, the C5 attached here and activates a series or set of complement proteins that are the C6, 7, and 8, yielding in a complex known as C5b, C5b, 6, 7, and 8. This complex stimulates the polymerization of 18 molecules of C9. 18 molecules of C9 are polymerized into a tube-like that penetrate the cell membrane of the microbial agents to form a channel. Through this channel, ions, small molecules, and water enter and out the microbial cell. Following to this process, lysis of the microbial cell occurs. This is the main aim of this reactions. Is microbial killing. Uh, this channel is known as MAC, membrane attaching complex. Now, the process of attaching of C5b into the cell membrane of the microbial agents and the following reaction is a process known as MAC formation. MAC, membrane attacking complex. This is the classical pathway. Now we will move on into the alternative pathway. In this pathway, there is a spontaneous conversion of C3 into C3A and C3B. C3B binds factor B to form C3BB. Now this complex is activated by factor D. This factor D yielding in C3BBB. This is an unstable complex and can be dissociated into C3B and BB. But this doesn't occur because this complex is stabilized by P protein or P factor, or known as propyridine, results in the formation of a complex, stable complex, C3b-PBB, which is the C3 convertase. Now, the C3 convertase acts on more C3, yielding in C3A and C3B. C3B binds to factor B, yielding in this complex, and activation of this complex occurs again, and the following reaction is repeated again and again. Now, when the C3B is accumulated, it binds to the C3 convertase to form C5 convertase, which is C3b, BB, C3b, C5 convertase. Now the C5 convertase acts on C5 to form C3a, C5a, and C5b. C5b attached to the cell membrane of the microbe, and the reaction repeat again, and microbial killing is achieved. Now the last pathway is the MBL, or menin binding lectin. This is triggered by the binding of lectin to menin. Lectin is a serum protein resembled to the C1q of a classical pathway. Now the lectin binds to menin. Menin is a manners of the bacterial cell wall, the viral envelope, and fungi. This binding will trigger the cleavage of two complement proteins, which are the C4 and C2, the same cleavage and same reactions of the classical pathway. But in this pathway, there is no activation of C1. This step is not considered with the MBL pathway. So this is the reactions of the complement system: classical pathway, alternative pathway, and lastly the MBL. It's all about the complement system. Thank you very much.